Good morning, everyone. Thank you very much for assisting to today's Xylella seminars. So I'm Luis, and I will be presenting today's speaker, who is Monica Donegan from the University of California. So Monica is a sixth-year PhD student in Rodrigo Almeida's lab, finishing her dissertation on Xylella fastidiosa. For her dissertation, she investigated local adaptation to temperature in distinct populations of subspecies fastidiosa within California. The first part of her research uses genomic methods to identify and validate temperature-associated genes, and the second part involves large-scale field experiments to test hypotheses about local adaptation. Today probably she will be speaking about this, so whenever you want, the floor is yours, Monica.
Okay, great. Thank you very much. You can hear me, everything. You can see the screen and everything. Okay, awesome. Then yeah, I'll jump right in. I'll be covering a lot of the work that I've done for my dissertation research today on Xylella. So I'd like to start with a brief introduction about what local adaptation is and how it applies to this bacterial system. So I think starting big picture, a pretty broad goal is to effectively manage the spread of Xylella under climate change. And to do this, a major method is to use species distribution models. And so species distribution models look at the current distribution of a pathogen like Xylella and use that to say what climate is suitable for Xylella, and then under climate change, what places will be suitable. So here's a map from a paper on Xylella that predicts climate suitability kind of across the globe. And so this isn't the range of Xylella, this is just theoretical, but to illustrate the example, what these models kind of assume is that populations of a species across a range have similar responses to rising temperatures. But this assumption may not be true because of local adaptation. And in local adaptation, different populations across a landscape already have different responses to temperature, and so may respond differently to climate change. And so, just to clearly define local adaptation, populations in a species are adapted for highest fitness in their local environment rather than their non-local environment. And so, if we think of the environment as a temperature or thermal gradient, and these populations in purple up north in California are exposed to colder temperatures, maybe these purple dots represent cold adapted populations, while the yellow dots represent warm adapted populations. And so here's what local adaptation looks like when you map fitness on the y-axis across a thermal gradient. And what I want to point out here is that usually in local adaptation, there's a fitness trade-off across the landscape. So there's not one population that does better in all environments. Instead, for example, this cold adapted population in the white squares has lower fitness than the other one when moved to high temperatures. So there's a trade-off across a landscape. Um, and so you're like, okay, I understand local adaptation, but the point is that not all species actually show local adaptation, so it's important to test for it. Um, there's some examples on the right of what these graphs of mean fitness looks like when there's no adaptation. The populations have similar responses to temperature, or there's not really a plastic response to temperature. And the point is that for a long time, people assumed that because a lot of microbial species have high rates of dispersal and migration, the populations were too connected, so it would be hard for populations to adapt to local temperatures. So there's a lot fewer studies of local adaptation to temperature, specifically in pathogens than in hosts. And so thinking about how this relates to Xylella, um, as we know, Xylella is transmitted by sap-sucking insects, and so the dispersal of the bacteria is limited by the insect. So that could create conditions for local adaptation. Um, Xylella is naturally competent, and we'll get into why that's important to the mechanisms of local adaptation later. And then today I'll be focusing on subspecies fastidiosa and Pierce's disease causing Xylella strains. Um, and so we'll focus on subspecies fastidiosa, but other subspecies will be relevant to our story. And so the reason we were interested in local adaptation to temperature specifically rather than other environmental variables was because of the strong impacts of temperature on Xylella fitness and growth. So on the left, we have an explanation of summer temperature impacts on Xylella population sizes. And so the timing of the inoculation into the plant, as well as how warm the summer is, can impact the growth of the population, right? And that impacts symptoms and epidemiology, and also the population size going into the winter before the plant is dormant. So summer temperatures generally lead to more growth, and colder winters lead to lower Xylella survival because of overwinter recovery. When there's colder winters, there's a higher probability of recovery. Mild winters, lower probability of recovery. So in general, this is an interesting, I guess, bacterial species to look at local adaptation because both summer temperatures and winter temperatures are strong, you could say, selection pressures on the fitness of Xylella. Okay, and then thinking about how local adaptation happens, um, there's two kind of main ways that we'll think about today. The first is vertical evolution. So this is just point mutation and selection on specific point mutations, right? And then I mentioned earlier that Xylella is naturally competent, and so there's the potential for horizontal gene transfer. So there's multiple forms of horizontal gene transfer. There can be lateral gene transfer where genes are kind of wholesale gained or lost, or homologous recombination where alleles are swapped between bacterial cells. And so this is interesting because we kind of want to know the relative impact of vertical evolution or horizontal evolution on how Xylella will adapt to climate change and how these two kind of evolutionary forces shape adaptation to temperature. Okay, and with that brief introduction, I'll get into some of my research results and I'll kind of go chapter by chapter of my dissertation. And so in the first part, I'll start with my kind of genomics results on showing on a genomic level that there's local adaptation to temperature by identifying genes and mechanisms. And then in the second part, I'll move to a pretty large scale field experiment of testing for local adaptation to winter survival, and then also estimating the rate of mutation in the field. And then finally, in the third chapter, I'll talk about a reciprocal transplant experiment where I'll get at what are the fitness trade-offs involved in local adaptation across distinct populations of Xylella. All right, so let's focus on the first chapter. And again, this one is mostly genomic and computational methods with some reverse genetics at the end. But to get into my research questions more, I first wanted to detect temperature-associated genetic variation. So genes and SNPs associated with temperature that could give a signal of local adaptation. And then I wanted to know how did these adaptive mutations arise, whether through vertical evolution or horizontal gene transfer. And then after identifying candidate genes that are associated with temperature, can we confirm these genes have a function in Xylella fitness, growth, cold tolerance, etc. All right, so to do environment and gene associations, you need a lot of genomic data. And previous members of the Almeida lab had sequenced about 130 subspecies fastidiosa strains from Sonoma County down all the way to Southern California. So these are all the black dots are whole genome sequences of Xylella isolates. And at the beginning of my PhD, I went to some of these more northern counties, so Mendocino County and then Humboldt County. And I added about 60 new isolates from this colder range edge. So these counties are slightly colder in the winter and Recently, Pierce's disease has been spreading here, and so we wanted to make sure we collected isolates from this colder area in California. And just to reiterate kind of what the temperature gradient is like in California, in general, in these northern coast areas, the winter is significantly colder than in other parts of the state. In the Central Valley, so in Kern and Fresno, it's very, very hot in the summer. And on these South Coast areas, so Riverside, Santa Barbara, San Luis Obispo, it's more temperate kind of year round. So the point is we have a large whole genome genomic data set with many SNPs differentiating these populations, and then also a temperature gradient representing different winter and summer temperatures throughout California. Okay, so first, before doing these gene environment associations, you need to make sure you understand the population structure and control for population genetic structure in your data set before association. So here's how our populations were separated in California. And what you'll notice is that in kind of northern areas, there's multiple genotypes circulating. So in Mendocino County, there's this teal and orange both circulating there. And then in Napa, this pink dot, and then in Southern California and Riverside, there's the same population. And so this is likely indicative of long distance dispersal events of how subspecies fastidiosa has been spread throughout the state. Okay, and then after running multiple gene environment association models, we ended up with a lot of SNPs in different genes associated with winter temperatures on the left and mean summer temperatures on the right. So for mean winter temperatures, focusing on the left first, there were 6 significant SNPs. Two of them were in this ROCF gene, which is a putative arginase, and then the other SNPs were in the kind of promoter region and region just upstream of this gene. So everything was in the same genomic region. And looking at an example of one of those SNPs on the map, the significant mutations were separating our populations in Northern California. So all of these purple dots in Northern California versus all of the Xylella populations in Southern California. So it's possible that these mutations in ROCF are adaptive in surviving winter temperatures. Okay, and then for summer temperatures, there were a lot more SNPs. There were over 200 SNPs associated with summer temperatures. And in general, these mutations separated populations in the Central Valley, so in Kern and Fresno counties. So these are the purple dots in the middle of the state in this yellow region where it's very hot in the summer from the rest of the population. So SNPs in these different regions throughout the genome may be helping these populations in the Central Valley adapt to very warm temperatures. And what we noticed in this Manhattan plot in the upper right is that a lot of the SNPs were kind of clumped in similar regions of the genome. So what we then did is look at if these regions that had a lot of SNPs were the result of homologous recombination using various computational pipelines. And overwhelmingly, of our about 213 temperature-associated SNPs, 98% were in regions that had inferred homologous recombination. So this was pretty surprising and a strong indication that horizontal gene flow is an important factor in temperature adaptation. So we then looked at these regions that had evidence of homologous recombination, ran some blast searches and computational pipelines to see where did they come from, how did they get into subspecies fastidiosa. And a lot of the sequences seem to be homologous recombination from subspecies multiplex. Multiplex has an overlapping range with fastidiosa in California. And if you look at a species tree, built from the whole genomes of all of these strains, fastidiosa and multiplex are very well-defined clades, right? But if you look at, let's call these gene trees of specific regions where there were many summer-associated mutations, what you see is that the sequences from strains in the Central Valley, so Kern County, have high sequence identity with subspecies multiplex strains. And so these strains from Kern County are subspecies fastidiosa, but rather than falling in these fastidiosa clades, they have a similar sequence identity with subspecies multiplex. And so at least for these genomic segments, there's evidence that there was horizontal gene transfer from multiplex into fastidiosa in the Central Valley. All right. And then separate from the mechanism of how local adaptation is happening, we wanted to know, are these genes actually involved in a function? So there was only one winter-associated candidate gene, this ROCF or arginase. And then for summer associations, there were 20 plus candidate genes. So based on a literature search, we chose three that were in different genomic regions. And I decided to make knockout mutants in two different Xylella strains. One strain, D06, is from Northern California and Mendocino County, so adapted to colder temperatures potentially. And one strain, JE115, from the Central Valley, that is exposed to very warm climates. So in general, I had two different strain backgrounds. I had wild type and then four gene knockouts in each of those strains. And I infected 10 vines, which with each of these 10 different strain constructs and then some control. So for all of these vines, I put them in the greenhouse and left them there for 12 weeks to look at symptom development and the population growth within the bacterial population growth. And then for the plants infected with wild type strains and my my knockouts of winter genes, I move them into a cold chamber for 6 weeks at 1.5 degrees, move them back to the greenhouse to see uh, overwinter survival after a very short cold exposure. Okay, and so looking at the results of symptoms on the left, we have area under the disease progress curve, so symptom aggregation over time. And on the right, we have the bacterial populations on the log scale, estimated from qPCR. What I want you to notice is that there were lower symptoms in these knockouts of summer-associated genes in both strain backgrounds. So in the D06 and in the JE115, there is evidence that knocking out these genes decreased symptom development. Populations were slightly lower in these knockouts, sometimes not significantly, but for some knockouts like this C1 peptidase gene, there were significantly lower populations than wild type. Um, so in general, there was evidence that these three summer-associated candidate genes impacted growth and virulence of Xylella in the plant. And just to show you some pictures in the wild type and the ROCF mutant, there were multiple leaves with scorch symptoms versus these summer gene mutants didn't have strong virulence in the plant. And moving on to our winter gene, this ROCF, what we saw is that when we put them in the cold chamber and brought them back out, we could see survival, and this is survival of the pathogen, right? And the first thing I want to point out is that the two strain backgrounds, the D06 from Northern California and the JE115 from Southern California, showed different winter survival. So JE115 in the purple bar, the wild type, actually none of the Xylella survived in those plants, and in the ROCF, there was some survival. So first, the strain background was pretty significant in determining survival. But yeah, when we knocked out this ROCF gene, survival from a brief cold exposure actually increased, which was not what we expected. And the populations were slightly higher in ROCF mutants than in wild type. Okay, so this was exciting because often you do these big computational models and then you're not sure what they actually mean, but we could demonstrate a function for multiple candidate genes. So these summer-associated genes have a function in growth and virulence in both strain backgrounds. And this winter-associated gene, ROCF, seemed to impact cold survival, especially in this southern strain background. All right, so hopefully I've demonstrated evidence of local adaptation in genomes, but then we want to move from genomes to actually testing it with field and greenhouse trials. So we want to show evidence of local adaptation, specifically with pathogen winter survival. And then also, because I had the opportunity to infect multiple different Xylella strains in the field, estimate the rate of mutation. Um, so in chapter one, at the end, I showed some evidence that JE115 and DO6 have slightly different survival in a short cold exposure. But I think everyone in this group knows that doing things in potted grapevines in controlled environments is very different than actually going to the field and infecting mature plants that are exposed to ambient conditions. And then the second part of this was because I could actually infect vines in the field, I wanted to know the rate of mutation. People usually do this with genomic data. So Alexandra and I, a member of my lab, estimated the kind of mean substitution rate using an ancient DNA sample of Xylella a few years ago. And other people have done this from looking at how modern strains are related, but it's hard to actually look at what the substitution rate is in the field. Okay. And the reason I could actually do this work was because there was this experimental vineyard in Hopland, which is in Northern California, that they were going to retire. In general, people don't want you to come to their commercial vineyards in California and infect all of their plants with Xylella. Totally understandable. This one was planted by a farm advisor who wanted to test Mediterranean grapevine cultivars in a California climate. So it had 13 different cultivars that are mostly planted in Europe, now in California. And all of these vines did not have Pierce's disease. We tested them before, but there was Pierce's disease circulating in Hopland. So we were given permission to infect this vineyard. And the setup of this was what's known as a common garden experiment. So we took two different distinct strains of Xylella subspecies fastidiosa, this DO6 from Hopland, and this JE115 from Bakersfield, and infected the vineyard that's in Hopland. So the strains are from two different climates, but they're being tested under the same climate and experiment location in Hopland. And what we expected going in was to see sort of a fitness trade-off between winter survival and then growth and symptoms, maybe during the growing season. So this is a common trade-off in bacteria because you have to allocate resources, right? So if you're allocating resources to survive stressful conditions, such as cold temperatures in the winter, there's fewer resources to allocate towards growth and plant colonization. So we hypothesized initially that this DO6 strain that's local to Hopland would have high winter survival, but the flip side would be it might be less virulent and show reduced growth in the plant, and vice versa for the JE115 strain. So we started this experiment in May of 2021 and then infected about 500 different vines we had controls with mock inoculations and then some with DO6 and some with JE115. And I won't talk about the variety part as much, but these were split up across 13 different cultivars. So there was kind of a host genotype and two different pathogen genotypes as well. And all of the vines were infected into the western quadrant of the plant so that we could track movement of the pathogen from the western quadrant across the vine as it spread. So this is looking at infections by strain before the winter, so in the first season in 2021. And in general, the infection success and the bacterial populations on the log scale, the middle graph, were not significantly different between the two strains. The local strain DO6 and the non-local strain JE115 showed similar results. But when we looked at symptom incidence, so in vines that were positive, did they show symptoms? There are actually more symptoms in vines infected with our local DO6 strain than the JE115 strain, which was counter to our hypothesis going into this. All right. And then we tested these vines many times, and I had kind of an undergrad army and many interns who helped me do many, many qPCRs throughout this project. And so we tested all 500 of these vines like multiple years in a row so we could look at overwinter survival. This is mapping overwinter survival of the pathogen. And so you can think of it as the reverse of winter recovery. So there was higher pathogen survival in our local strain. And this was true after one winter and after two winters. The second winter is graphing, or the second part of the graph on the right is not cumulative. It's of how many vines that were still positive in 2022, how many recovered. So what I think you can see is there's a very strong trend. And this is pretty high sample size, right? We had a 500 vine vineyard. And so this is a very strong kind of signal that there's local adaptation to temperature. Another thing I want to point out is that there was actually lower survival after 2 years from 2022 to 2023. It was a much colder winter that year. And so obviously the environment has a pretty strong impact on recovery or pathogen survival, and many other things that I'm not going to focus on today, like the pathogen population size before the winter, as well as variety. And then looking at our symptom incidence after the winter, this is just in positive vines, so it's independent of whether the vines recovered or not, and that DO6 survived at higher rates than JE115. So separate of that, JE115 showed lower symptoms than DO6 kind of throughout the trial and on all quadrants of the vine. So you can see in 2022, there was some spread across the vines, and in 2023, there was a lot of spread, but there were always more symptoms in vines infected with DO6. All right, and then to look at what the mutation rate was in the field, I recultured strains. So I'll talk about these as my derived isolates. There were two parent strains, the JE115 and DO6, and I had some derived isolates from one field season in 2021, and then many more derived isolates in 2023 after three field seasons. So I did a lot of computational work to kind of map the different strains back to a reference strain and back to the parent strain to figure out where mutations had occurred in the field evolved strains. And using this, I was able to estimate the substitution rate, so mutations per site per year, in both strains across 1 and 3 field seasons. And broadly, it was concordant across the strains and across the two time points. But in general, the substitution was a little bit lower than when estimated from genomic data, which may indicate heterogeneity across the landscape. Because I had all of these mutations, I wanted to know not just what's the rate of mutation, but also are there any interesting mutations? So first, I made sure that all of my derived strains matched my parent strains, right? Like what I put into the vines was what I thought it would be. And then I looked at where mutations were occurring throughout the genome. And there were actually a ton of mutations in this type 4 pili operon. So these genes are minor type 4 pillins, pill W1, pill X1, and pill E1. And there was kind of a crazy amount of convergence of strains that had mutations in these genes after they had been evolved in the field. So these lines are showing the number of derived strains that had a specific mutation, and they're color coded by the parent strain. So purple lines are DO6, red lines are JE115. And so by the end of the trial, I had 43 derived isolates, and 42 of them had a loss of function non-synonymous mutation that was messing up the protein in one of these genes. So this was kind of crazy. And I did a lot of work, kind of had a big PhD rabbit hole to validate that this had actually happened in the field by doing a lot of PCRs and Sanger sequencing and hadn't happened in the lab before I put these into the field. Um, so I kind of went to the literature and looked at what the function of these knocking out these minor type 4 pillins were. And so they're predicted to have no twitching motility, so the fringe width is zero, and they are not able to recombine. And so a grad student in Leo's lab at Auburn, Mamata, helped me out by running some twitching motility assays. And we looked at twitching motility in the parent strains of JE115 and DO6, as well as some derived mutants. And what we found is that none of the derived mutants, so strains that had mutations in these PIL-W or PIL-X genes, had twitching motility. But interestingly, in the JE115 parent strain, there was twitching motility. You see this nice fringe around the colony. But the DO6 parent strain already didn't have twitching motility. So this was kind of weird because there were still all of these mutations popping up in these genes in the DO6 genetic background. And so it may be that twitching motility is not the sort of phenotype under selection because there was still selection for mutations in these genes in DO6, which already didn't have twitching motility to start with. So then I also tested whether the derived mutants still were able to cause symptoms in the plants, and broadly they were. So this was a little bit of a puzzle that I'm still kind of thinking about. And again, looking at what happens when you fully knock out these different genes. For some of these knockouts, they actually have increased growth relative to the wild type. And so this could be an advantage in having faster growth before the winter to better survive colder winter temperatures. But the interesting thing is that in general, subspecies fastidiosa strains retain twitching motility in nature, and you don't see mutations in these minor pillin genes that I saw were highly, highly under selection in my field trial. And so there can be trade-offs, kind of what we see in nature versus what we see in this experimental evolution. Setup, especially with the insect acquisition phase, I mechanically inoculated all of these vines. And so there could be kind of trade-offs with that that explain why we don't see these pill mutants in nature. But there is evidence that they have some sort of function and that when you put them into the vine in this colder climate, there's some sort of adaptive evolution for removing twitching motility or having mutations in these genes that cause faster growth. Okay, and then I will talk about my final chapter where I look at fitness trade-offs involved in local adaptation to temperature across distinct populations. And so my experimental setup in my second chapter was a common garden. And allows you to answer, does the local strain outcompete the non-local strain? And our answer was yes, right? The local strain DO6 had higher winter survival and it also had higher symptom incidence in infected vines. Um, a reciprocal transplant is when you take those distinct genotypes from two different places and then you move them to those respective places or you to the reciprocal places. And what this allows you to ask is, does adaptation to one environment have a cost in another environment? So we're getting at trade-offs rather than just does this local strain beat the non-local strain in one environment. We're testing it in both environments. And so I set up my reciprocal transplant experiments using multiple strains from each genetic cluster rather than just DO6 and JE115. I had 5 strains from Hopland. I'll call these my Hopland cluster or HCC strains, and then 5 strains from Bakersfield, the Bakersfield cluster strains. And so I think you're well acquainted with these are different climates, and also these strain clusters are genetically distinct. And first, I looked at some in vitro phenotypes, growth under different temperatures. And then I ran these reciprocal transplant experiments. And I did 2 experimental replicates of this whole experiment. So I ran one with 200 plants from 2023 to 2024. And then I repeated the whole thing from 2024 to 2025. And what I did is I had 200 plants in the greenhouse at Berkeley infected with Hopland cluster and Bakersfield cluster strains. I transported them to the field sites for the winter. So half the plants went to Hopland in these screened in tents for the winter. Half the plants went to Bakersfield for the winter. After the winter, in mid-April, I moved them back to the greenhouse so I could look at pathogen winter survival as well as symptoms after the winter. Okay, so another grad student in Leo's lab helped me run all of these. So Ranlin uh, did most of this work. And what we did is look at the Hopland cluster strains. So there were 5 strains, and Bakersfield cluster strains, like also 5 strains, and their growth at 20 degrees and 28. So what was interesting is at 28, they grew very similarly, but when you grew them at a lower temperature at 20 degrees, the strains that were from a warmer climate actually grew faster at a colder temperature. So it's a little bit counterintuitive. Um, but these warm climate strains have faster growth at 20 degrees. They also had more biofilm and planktonic growth at 20 degrees, but the strain clusters were not different at 28. So we didn't test at higher temperatures, but kind of our inference from this is that the thermal optimum of these Hopland cluster strains from a colder climate is a little bit increased. So they may need to grow rapidly at higher temperatures, but the Bakersfield cluster strains have a slightly lower thermal optimum to potentially restrict their growth. And I'll get into why later. Um, so then moving from the in vitro work into the reciprocal transplant work, over experiment 1 and experiment 2, it was colder in Hopland than Bakersfield, which it better be because that's kind of a baseline assumption of most of my work. And in the second winter in experiment 2, it was colder than in the first year. All right, so I'll first talk about pathogen winter survival, and I'll split this up by grapevine variety because in the first experiment I used all Cabernet Sauvignon, and in the second experiment I had to use half Cabernet Sauvignon and half Xylella, um, which is one of the Mediterranean cultivars that I had from my field trial. So looking at the first 2 boxes in experiment 1 and experiment 2, in Cabernet vines, when you move from a warmer climate, Bakersfield, to a colder climate, there's lower pathogen survival of these Bakersfield cluster strains, right? So these warm climate strains don't survive as well under a colder winter. This result is pretty concordant with our field results. In Chile Jolo, I had this kind of crazy result where the warm climate strains just don't survive very well under either climate. So this red line at the bottom. Um, and meanwhile, these Hopland cluster strains survive at 100% regardless of the climate. So I think the point is that there's very strong host and pathogen genotype interactions here, right? But overall, these Bakersfield cluster strains had lower winter survival than the Hopland cluster strains. Then I looked at how the infected vines broke bud after the winter. And what I noticed in the first experiment was that vines that were infected with these cold climate Hopland cluster strains were very unhappy after the winter, especially the plants that had overwintered in a warm climate in Bakersfield. So this purple line that's solid are the plants infected with Hopland cluster strains in Bakersfield. And you can see there was pretty delayed bud break in the spring. And some vines actually never broke bud. They were completely dead after the winter. In the second experiment, which was slightly colder, there was still an effect where plants infected with cold climate strains broke bud later, but there wasn't an effect of location, possibly because it was colder in both places. And just to give you an additional sense of what this looked like, at the end of the growing season after the winter, so after the plants had been put in the field and brought back, I looked at scorch, severe stunting, and vine mortality. And scorch wasn't different between the 2 strain clusters, but stunting and vine mortality were both higher in vines infected with these cold climate Hopland cluster strains and slightly higher when they were put in Bakersfield than in Hopland. So in a warmer climate, there was more symptoms after the winter. Okay, and then I started off with talking about this survival growth trade-off, but pretty much that got thrown out the window after the field trial, and we were back at the kind of drawing board to figure out, well, what is this fitness trade-off in local adaptation? So what we were seeing is that these northern strains, these hopland cluster strains, have not only greater winter survival, but they seem to have higher virulence and potentially faster growth at high temperatures. And so it could be that there's a trade-off between virulence and transmission, and that when you move these hopland cluster strains to a warmer environment, they are too virulent and there may be lower transmission and pathogen spread across the landscape. And the reason we thought this could be true is because insect vectors of Xylella discriminate against plants with symptoms. So past work has shown that if given the choice between infected or healthy plants, they prefer to feed on healthy plants. And so if plants have symptoms, especially after the winter too early, like this severe stunting or vine mortality, that may not be advantageous from Xylella's perspective. And so there could be like a trade-off between virulence and transmission and therefore overall pathogen fitness. So putting this together in a conceptual model, this is just what we think is happening, not the actual data of pathogen fitness that we measured, but um, I think pretty consistently there is data that the warm climate strains have lower survival in cold, right? And especially when you move them to a colder winter, they have lower survival than the cold climate strains. So looking at this red line, there's a decrease in fitness when moved to a colder winter. But our hypothesis for a drop in fitness in these cold climate strains is that when they're moved to a warm winter environment, they're actually too virulent and they may cause very severe symptoms after the winter. And that's negative for fitness because it can impact transmission and then therefore pathogen spread across the landscape. Okay, I'm going to bring it back to my genomic work that I presented at the beginning of this and talk about why homologous recombination from subspecies multiplex might be adaptive, specifically in these warm climate strains in the Central Valley. So multiplex is in general found in colder regions than fastidiosa. It grows more slowly in vitro and it doesn't really show virulence in grapevines. Some strains do, but in general, there's lower virulence in grapevines. And so it could be that horizontal gene transfer of these multiplex alleles into fastidiosa populations in very hot climates is actually adaptive because it helps them to modulate their virulence and to kind of restrict their growth a little bit to not cause too many symptoms, which could impact transmission and pathogen spread. And to kind of sum up all of my conclusions from my dissertation work, first, in chapter 1, we looked at molecular mechanisms of local adaptation, and there was multiple candidate genes that were validated that have impact on Xylella virulence and cold tolerance. And we found that horizontal gene transfer, especially from subspecies multiplex, was contributing to local adaptation. And then moving into my field and greenhouse work, there was strong evidence of local thermal adaptation in winter survival in a pretty large field trial. And then using a reciprocal transplant experiment, there seemed to be a fitness trade-off across populations. And we hypothesized that this is about virulence and hypervirulence when moving cold climate strains to a warmer climate. All right, so yeah, I've been at Berkeley for 6 years and I've gotten to interact with many wonderful mentors and collaborators, and I had just a ton of help with all of these projects because I did a lot of field work and lab work. I had, yeah, many students who helped me who were really wonderful in making all of this possible. And yeah, I dragged many of my lab members to different places in the field with me, and I got to travel with them a lot, and it was a great time to spend time with them. And I'm happy to take questions now. I also have my email if you'd like to talk about anything more. I'm very open to feedback and would love to get everyone's thoughts on this work.
Great. Thank you very much for your very interesting presentation, Monica. It was very insightful. So for anyone in the audience, please feel free to ask questions to our speaker.